Welcome to aminacademy.com. Let us do in human anatomy and physiology. We will discuss about the cell cycle. Many damaged, dead, and worn out cell can be replaced by growth and division of their similar cells. The frequency with which cell divisions occurs varies with different type of tissues. This is normally carefully regulated to allow effective maintenance and repair of body tissues. At the end of their natural lifespan, aging cells are programmed to self-destruct and their components are removed by phagocytosis. This process known as apoptosis. Cell with nuclei have 46 chromosomes and divide by mitosis, a process that results in two new genetically identical daughter cells. The only exemption to this is the formation of gametes, that is sex cells, ova and spermatozoa, which takes place by meiosis. The period between two cell divisions is known as the cell cycle, which has two phases that can be seen on light microscopy: mitosis and interphase. The human cell cycle is process by which the cell replicates its DNA and divides into two daughter cells. The cell cycle has four main phases. Gap 1, that is G1, the cell grows and checks that it's really to divide. Synthesis is the cell copies its DNA. GP2, that is G2, the cell checks that its DNA has been copied correctly. Mitosis M, the cell divides into two daughter cells. That's explanation. The cell cycle is similar to a life cycle where a cell grows and develops from its birth to its reproduction. The cell cycle main function is to create two genetically identical daughter cells. The length of each phase varies between the different types of cells. For example, a rapidly dividing human cell might take 24 hours to complete the cycle, while early embryonic cells might take 30 minutes or less. The cell cycle ends when the daughter cells each have their own outer membrane. Next, we'll talk about cell cycle in that interphase. What is interphase? This is the longer phase and three separate stages are recognized. First, gap phase, that is G1. The cell grows in size and volume. This is usually the longest phase and most variable in length. Sometimes cells do not continue around the cell cycle but enter a resting phase. During this time, cells carry out their specific functions. Example, secretion, absorption. Next one, synthesis of DNA. The chromosomes replicate, forming two identical copies of DNA. Therefore, following the S phase, the cell now has 92 chromosomes, enough DNA for two cells, and is nearly ready to divide by mitosis. Second gap phase, there is a further growth and preparation for cell division. In the human cell cycle, interphase refers to the lengthy stage where a cell grows, replicates its DNA, and prepares for cell division. Essentially encompassing the majority of the cell's life before entering mitosis, cell division, where it splits into two daughter cells. It consists of three subphases: G1, that is gap 1, S synthesis, and G2, gap 2 phase. Key points about interphases, how it's happened. Major function. During interphase, the cell actively grows, synthesizes proteins and other molecules needed for division. The most importantly replicates its DNA in the S phase. Subphases. G1 phase, the cell grows and prepares for DNA replication, performing its normal functions. S phase, DNA replicates occurs, creating identical copies of each chromosomes. G2 phase, the cell continues to grow and synthesize proteins needed for mitosis. Now we'll talk about mitosis. This is a continuous process involving four distinct stages visibly by light microscopy. Prophase. During this stage, the replicated chromatin becomes tightly coiled and easier to see under the microscope. This each of the original 46 chromosomes called chromatid at this stage is paired with its copy in a double chromosome unit. The two chromatids are joined to each other at the centromere. The meiotic apparatus appears. This consists of two centroids separated by the mitotic spindle, which is formed from microtubules. The centrioles migrate one to each end of the cell, and the nuclear envelope disappears. Metaphase. The chromatids align on the center of the spindle attached by their centromeres. Anaphase. The centromeres separate and one of each pair of sister chromatids, now called chromosomes again, migrates to each end of the spindle as the microtubules that form the mitotic spindle contract. How the mitosis occur? Mitosis is a phase that occurs in human cells to create two identical daughter cells. It is fundamental process of growth, development, and repair. How does mitosis occurs? Look at this figure. The cell DNA replicated, the cell's chromosomes condense and pair up, the cell nuclear envelope breaks down, the cell's centrosomes organize and meiotic spindle. The meiotic spindle pulls the sister chromatids apart. The cell's cytoplasm divides into two daughter cells. Why the mitosis important? This is very important in cell divisions. Mitosis ensures that each daughter cell receives an identical set of chromosomes. Mitosis is essential for the growth, development, and repair of the human's body. When mitosis is not regulated correctly, health problems such as cancer can result. Finally, we talk about what is cell cycle. A cell cycle is a series of events that takes place in a cell as it grows and divides. The cell spends most of its time in what is called interphase, and during this time it grows, replicates and chromosomes and prepares for cell division. The cell then leaves interphase, undergoes mitosis, and completes its division. The resulting cycle, known as daughter cells, each enter their own interphase and begin a new round of the cell cycle. The cell cycle is the name we give the process through which cells replicate and make two new cells. Cell cycle has different stages called G1, S, G2 and M. G1 is the stage where the cell is preparing to divide. To do this, it then moves into the S phase where the cell copies all the DNA. So S stands for DNA synthesis. After DNA is copied and there is a complete extra set of all the genetic materials. The cell moves into the G2 stage where it organizes and condenses the genetic material or starts to condense the genetic material and prepares to divide. The next stage is M. M stands for mitosis. This is where the cell actually partitions the two copies of the genetic material into the two daughter cells. After M phase completes, cell division occurs and two cells are left, and the cell cycle can and cell cycle continue. So far we discussed about human cell cycle some detail. We have done. Please subscribe our YouTube channel.